Before we start talking about conjugation, our next form of horizontal gene transfer that the prokaryotes can do, we just need to remind ourselves about the plasmids again. So remember that plasmids are just pieces of DNA that are usually found outside the bacterial chromosome, and they contain genes that give the bacteria an advantage. So we've got some different types of plasmids. So the one that we're going to be talking about with conjugation would be these conjugative plasmids. They're also called F plasmids. And these conjugative or F plasmids contain genes that code for how to make a sex pilus. And the sex pilus is going to be used in conjugation to build a little protein tube to transfer over a copy of the conjugative or F plasmid to the other bacterial cell. And then the other type of plasmid that is a concern in conjugation would be these R plasmid or resistance plasmids. So these plasmids contain antibiotic resistance genes. And through conjugation, this is another way that the bacteria can spread antibiotic resistance genes from one bacterial cell to another. All right, so conjugation is just going to be when two bacterial cells are going to be in direct contact with each other through this little hollow protein tube here called a sex pilus. And what's really scary about conjugation is that you can see with this picture that these two bacterial cells are not even the same species. So conjugation can happen between two bacterial cells that are not even the same species. And as they transfer over from one bacterial cell to the other a copy of the F plasmid or the conjugative plasmid, that F plasmid can actually contain some antibiotic resistance genes. And so we can take antibiotic resistance genes from this one bacterial cell and then spread them to the other bacterial cell. So we can use horizontal gene transfer with this conjugation as another way to spread antibiotic resistance genes from one bacterial cell to another. So the reason that the conjugated plasmids are also called F plasmids. F here just stands for fertility because they thought that conjugation sort of looks like the bacteria doing sexual reproduction, but they're actually not. So that's just where that name came from. All right, so let's just take a look at what's going on in conjugation. So we've got a couple of terms here. So the bacterial cell that has the F plasmid or the conjugative plasmid, that one's going to be called the F plus bacterial cell. Okay, so the one that has the F plasmid is called the F plus bacterial cell. And it's only going to do conjugation with an F minus bacterial cell. So it will only do conjugation with a bacterial cell that does not have an F plasmid. And so this F plus bacterial cell will come over, find an F minus bacterial cell, and that F plasmid will code for the proteins to make this sex pilus. And then this sex pilus will form this hollow tube here and join the two bacterial cells together. And then once they're joined together, we're going to nick one of the strands of the double-stranded DNA in the plasmid of the F plasmid here, and then we're going to send over one of the strands of the F plasmid and give that to the F minus bacterial cell. And now when we're done doing conjugation, both of the bacterial cells will now have a copy of the F plasmid. And so now they're going to be both F plus cells. And so now this bacterial cell that was F minus is now F plus, and it has now gained the ability to do conjugation because it now has a copy of that F plasmid. And that F plasmid may have antibiotic resistance genes on it. And now maybe we've shared some antibiotic resistance genes from one bacterial cell to the next.